Alright class, so now these last few videos are going to be looking at different kinds of inheritance and genetics problems. Okay, so again we're kind of starting here with sort of the basic, simple, what's the easiest kind of inheritance we can have, and that is something called complete dominance. Okay, so complete dominance means that if a dominant allele is present, right, so if a dominant allele is present, it is always expressed. If a dominant allele is present, it is always expressed as the phenotype. It's basically, it's a way of saying that as long as we have one dominant allele, we're going to see the dominant trait. That's what this is really telling us, right? As long as we have one dominant allele, that means we're going to see the dominant trait. Okay, for this list of problems, we're going to have dominant alleles and recessive alleles, and we're going to have dominant traits and recessive traits. So there's two possibilities for each, and when we're looking at the traits part of this, to see the dominant trait, all we have to have is one dominant allele. It doesn't matter what the other allele is, as long as there's one dominant trait. So that's complete dominance, right? The dominant allele has complete dominance over the recessive allele. Even if they show up together, we still see the dominant trait. All right, so that's our sort of description of what complete dominance is. All right, so on our first problem here that we're working, we're going to go with a character of flower color in pea plants. So we'll kind of do one of Mendel's experiments or crosses here. So flower color in pea plants. Notice not specific, right? It's just the general feature that we're looking at. All right, the dominant trait is purple flowers. The recessive trait is white flowers. Okay, so there's some information here that you have to be given, right? Like it's no good for me to just say cross this with that, because then it's like, well, am I doing complete dominance, or like what's the character? What are my traits? Like, you don't know all that stuff, and you have to know all that stuff. So in any problem you would be working, I would be telling you things like, this is complete dominance. Here's the character, here's the dominant trait, here's the recessive trait. I would be telling you those things. Okay, and then I would also, just for the sake of consistency, I would tell you the letter really doesn't matter, but I would just say something like, use the letter A. So that it's just kind of consistent, and most importantly, uppercase, lowercase are easy to tell apart. Because, been doing this a long time, so you end up in a Punnett square where somebody picks their own letter and they pick S. So then they just kind of do this on their Punnett square, and it's like, what the hell are those? I mean, yeah, they're S's, but I can't tell what's what. So unless you like really exaggerate it like this, but I mean, that's too much work. So, if you use A's, B's, D's, G's, H's, whatever, right? Those are things I pick to use that make it easy to do the Punnett square. All right, and then on this first cross, what we're going to do is cross homozygous dominant going to cross homozygous dominant with homozygous recessive. Homozygous dominant with homozygous recessive. So I've told you, use the letter A in our crosses. So we want to go back now and write in our genotypes. So homozygous dominant using the letter A. And we're crossing that with homozygous recessive using the letter A. Right, so all we've done is rewrite our cross using our shorthand letter way of doing genotypes. And this is the P generation. Remember from the pre-lecture stuff, like, oh, my lecture videos so far, I've just kept calling this the parent generation. But the shorthand for that is P. So these are our parents. These are the P generation. All right. So remember, then next step is going to be gametes. Right. What my gametes are going to be for each parent. Hopefully by now from watching the stuff we've done, you know how to do this step. We were doing this in person in class. I would be telling you, if you don't know how to do this step, you should stop me and ask now, because everything else we're doing through the rest of this chapter is going to do this. We're going to be using this step. So email if you're not sure. But there's our gametes. And then we have a 2x2 Punnett square. Everything in the beginning here is just one character, so 2x2 Punnett square. Gametes for one parent for each row, other parent for each column, and then we combine columns with rows. Remember, it's technically not wrong if you put little a then big A, but it's just harder to interpret, especially in the beginning. It's easier to see that big A first. Right, so then remember that these genotypes inside the Punnett square are our possible offspring genotypes. And in the pre-lecture assignment, I had you write, there's an abbreviation we use for offspring generations. It's an F with the subscript of a number. So F1, F2, F3, F4, and they go in order. So if we started with our parents, then what's inside this Punnett square would be called the F1 generation. Parents make F1, F1 makes F2, F2 makes F3, F3 makes F4, right? So it's just numerically in order now. All right, and then there's a couple of questions we're going to ask about this. We're going to ask you to do what's called a genotypic ratio. So we're going to look at the genotypic ratio of the F1 generation. Hey, and you've heard me say this a couple times already, but this is really important. This is another step where a lot of people will be like, dude, I'm not writing that every time. That's stupid. But it helps you get correct answers. And nothing you can do to help you get correct answers is ever stupid. So genotype. We have 3 possible genotypes. Forget looking in the Punnett square right now. Just think we've got 3 things we might see. Those are them. We might see big A, we might see big A, we might see little a. Those are all of our possibilities. Now we can go back to our Punnett square, F1, look up here. In those Punnett squares, how many are big A, big A? 0 out of 4. And again, I know some people may be like, well, dude, it's 0. It's stupid. I'm not putting that there. But I'm telling you, you're going to miss some questions if you don't do it this way, because you're just going to be like, well, I don't know what you mean. And this is the easiest way to keep track of everything. Right? How many are big A, little a? 4 out of 4. Little a, 0 out of 4. So that is our genotypic ratio for our F1s. And then we also want to do a phenotypic ratio. All right, so remember phenotypes, what we see. So in this case, phenotype is referring back to these things here: dominant trait, recessive trait. Right, so purple's dominant, white is recessive, and we're doing complete dominance. Right, so our options here are going to be purple and white. And we know, because this is complete dominance, we know that anything that's got one dominant allele at least is going to be purple. Right, so purple, 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 purple. 4 out of 4. 0 out of 4 white. Right, so that's the phenotypic ratio. We have to go back to our initial, what are our two traits, purple and white, and the idea that it's complete dominance, so that as long as we see that dominant allele, we're going to see the dominant trait, which is purple. All right, I'm going to show you one more example here with this, and then you're going to have a few to work on your own with the quiz that goes along with this. So now we're going to take our two F1 offspring. All of our F1s were heterozygous. So these are F1s. And we're going to cross them. Okay, so what are the gametes? Big A or little a, big A or little a. 2 by 2 Punnett square. Gametes for one parent, each row, gametes for the other, each column. Fill it in. Right, so now this Punnett square is F2 offspring. I mentioned to you earlier, parents make F1, F1 makes F2, F2 makes F3, etc. So we use two F1s here, right? These were F1s. The parents for this cross were F1s, so we're making F2s. Okay, and then let's do our two ratios again. So we're going to have a genotypic ratio of the F2s and a phenotypic ratio of the F2s. We're going to list all our possible genotypes and then go back and count. Big A is 1 out of 4. Big A, little a is 2 out of 4. Little a, 1 out of 4. So there's our genotypic ratio for our F2s. And then our phenotypic ratio was purple to white. Right, so purple was anything that has at least one dominant allele. Right, so this is purple, this is purple, this is purple. The little a is not. Right, so 3 out of 4 purple, 1 out of 4 white. All right, and then one last thing with this. This is the basic complete dominant stuff. So like when you see problems, you're going to have to work on this for understanding it. It's not going to be the same stuff we've did, right? It's going to be, here's a different character, here are the different traits. It may be using letter B or G or H, it doesn't matter. The parents, which you're crossing, may be different. So the questions you're going to see with the quiz related to this video is going to be, do you understand how to do this? You know, you can watch this thing a million times. You can practice with it, quiz yourself, email me if you have questions, that kind of thing. All right, and I want to mention real quick the idea of like why do these ratios in fractions? Because a lot of times if you look at biology books, they'll have these written just as ratios. Like for this, they may just say 1 to 2 to 1 as your ratio, and that's okay if you've done enough of this to really understand what that means. But if your answer is 1 2 1 and you have no freaking clue what it is, that does us no good. So that's why I just stick with the fractions in our ratio. And why that helps is, remember I mentioned when you do a cross, like you're not going to get 4 offspring, right? These are pea plants. So you can have a whole entire pea plant full of flowers that make peas, and the seeds, the actual round peas, are the offspring. So it's not making 4, right? A pod of peas has, 5 or 6 or 8 peas in it. So you can get offspring generations like an F2 that have thousands of offspring. So if we were saying we actually did this experiment and we had 3,290 F2 offspring, right? If we knew that from actually doing the experiment, and somebody came up and said, well, you had that many offspring, how many are going to be heterozygous? Well, we can know that because we could take 2/4 times 3,290, and that would give us how many we would expect to see being heterozygous, right? Or homozygous recessive, 1/4 times 3,290, right? If you had that 1 to 1, well, what are you going to do then? You're going to go, oh, well, how many are heterozygous is 2 times 3,290. So twice as many as the real population is going to be heterozygous. What? Right? So that's like, that makes no sense. That's what you're going to do if you use that ratio. That's the easy, let me write this fast way of doing it. So the fractions help us with, if this is how many offspring we really have, what are the numbers we would expect to see?